All right, this video is Unit 6 EOC Review Genetics Part 2. I'm going to be doing um, Mendelian genetics, things like Punnett squares, that sort of thing. Gregor Mendel was a priest, was a monk, was also a naturalist in that he wanted to test some different things. He started breeding pea plants and bees, and from his work, he found out these very simple principles of genetics that have stood the test of time. And so we're going to look at the three principles of Gregor Mendel real quick. First is the principle of dominance, and this is just that there are two alleles. There are dominant alleles and recessive alleles. And dominant alleles always express themselves, always show themselves, whereas recessive alleles can only express themselves if there's not a dominant around. All right, there's the principle of segregation. All right, and the principle of segregation says this, that any organism has two chromosomes, say, or every organism has two copies of a gene, is a better way to say that. And let's say that's their genotype, which is just their genetic makeup. That organism, when it's making gametes, has an equal chance to pass the big T as it does the little t. We often show this by doing something called a Punnett square. So let's say that that organism mates with another organism that has a similar genotype. Well, if they didn't separate, then we would only expect to ever see the dominant trait. Let's say that's tall in this case. But because they separate, there is a chance that that recessive trait will come back out, thus creating the recessive allele or the recessive phenotype, which is the physical expression. In this case, that would be short. Now, Mendel's third principle is that of independent assortment. And this has to do with when you're dealing with more than one trait. So let's say um, A and B here, that these traits are not necessarily linked together, meaning that just because you have one trait, A, big A, let's say big A is tall, little a is short, big B is brown hair, little b is blue hair. I don't know why you have blue hair. But you're not always going to have tall brown hair people. You could have tall blue hair or short brown hair or short blue hair. This one individual is able to produce, because of independent assortment, four different kinds of allele combinations. So how does this work itself out into the world of probability and Punnett squares? Well, really simple. The simplest kinds of Punnett squares are just the ones like I did earlier, where you fill them in. Of course, and this isn't the hard part. The hard part is actually coming up with the problem. And so let's just say that there's an individual that is homozygous dominant for tall. So what would that mean? Well, homozygous means same, homo, same. Homozygous dominant, so he's dominant and the same. So there's going to be two big T's. So homozygous dominant for tall is mated to someone who is heterozygous. Hetero means different, so hetero is two alleles that are different from one another. what is the chance that they would have an individual that is also heterozygous? Well, how do you do that? You can make a Punnett square. This is the kind you can do in your head, and you can see, yes, that is indeed a 50% chance. Now, another way to figure that out is through the rule of multiplication. Now, what does that mean? Well, if I want to figure out what is the percent chance that this will happen, I can simply do a little bit of multiplication of fractions. And so let me show you. Take that same cross, and we're trying to figure out what is the chance that this will happen. Well, what is the chance that this person here is going to donate a big T? 100% chance they're going to donate a big T. What is the chance that this one is going to donate a little t? Half. Multiply those two things together, you get 50%. I didn't have to do my Punnett square to do that. Now, on a problem like this, you may just want to do a quick Punnett square in order to say, make sure you're right. However, if you're given a problem like this, and you're told to give me the percentage of chance that the organism will have that genotype, then we have a problem. And so you need to do some quick math to do this, or you can spend 20 minutes on that question, which I don't recommend at all. So how do we do that? Well, we're going to consider each set of letters separately. What is the chance that this can be made from this and this? Well, again, if we did our little Punnett square, we would see that that's a 1/4 chance. All right. So we'll come here, we'll put 1/4. Well, what's the chance that this and this will make this? And again, we could do our Punnett square, but we would find out that the chance is 1/2, or 1 and 2. And so now we just multiply those together, and we see that it's a 1 in 8 chance, or what would that be percent-wise? It'd be like 12.5%. Let's do another one. What is the chance that an organism with this genotype will come about? Well, little a is 1/2. Big B is also 1/2. And so you have a 1/4 chance that that genotype is going to show up. And so that's the basics on Punnett squares. Now there's a couple of Punnett squares that don't fit the rules that we've talked about. One of them is called codominance. And codominance is when two alleles share dominance. And so let's say that you have a white cow and a brown cow, and both of those are dominant. And the brown cow, homozygous dominant brown cow, mates with a homozygous dominant white cow, and you get a mix. Well, in this case, because they share dominance, both of them are going to show up, and so what would you expect to see? You're going to have a cow. This is going to be the worst cow that's ever graced a page of anything. But this cow is going to have spots. It's going to show both white and brown. All right. So yes, horrible cow. Now there's also incomplete dominance. And this is where the organism or the trait doesn't completely dominate. So let's take red flowers are dominant, as are white flowers. Well, you're going to get not any bees. You're going to get this mix in all the squares. In this case, instead of having spots, you're going to have flowers that are pink, neither red nor white, because white and red are neither completely dominating. Now, a couple other situations, you can have multiple alleles where you have one trait that can have multiple alleles, and whereas this trait was white and red flowers, let's say that we had white, red, blue, orange, purple, all right, in one trait, color. Well, there would be a hierarchy of dominance in that one thing would be more dominant than the other. There'd be one that was the most dominant or the most recessive. You can have a situation where there's a polygenic trait. This is one trait, say skin color, and multiple genes can code for that trait. Now, I don't suspect that you'll see any problems like that, but incomplete dominance and codominance could both rear their heads. Now, lastly, is this concept of sex-linked or X-linked crosses. Now, why is this a special thing? Well, sex chromosomes are different from autosomes. Autosomes are just your normal chromosomes. Sex chromosomes are either your X or Y chromosomes, whereas the female has two X's and the male has an X and a Y. Well, why is this different? Well, for a gene like color blindness, whereas normal is dominant and color blindness is recessive, a female carries two alleles. In this case, this female's homozygous or heterozygous. She's normal. But the male only carries one allele because the Y chromosome does not have the allele for color blindness. So if he has one recessive allele, then he is colorblind. So let's take this pair that we just made and make a Punnett square with them. Now, when you do a Punnett square like this, the first thing I like to do is go ahead and fill in my X's and Y's. What does this demonstrate? This just demonstrates the probability of a cross between male and female, the chance that there's going to be males, and the chance that they're going to be female, which is 50-50. Well, this female is heterozygous for the color blindness trait, whereas dad is colorblind. Well, what is the probability that they'll have a colorblind daughter, say? Well, mom always gives her X chromosomes to her son. Or the sons always get their X chromosomes from their mom. Mom gives X chromosomes to her daughter, too. Dad gives his Y to his son, but he always gives his X to his daughters. And so there is a chance that this couple could have a colorblind daughter. There's also a chance that they could have a colorblind male. But there's a 50% chance that they're going to have a kid that's normal, and a 50% chance that they're going to have a kid that has color blindness, either male or female. Now, one last thing with these Mendelian genetics is this concept of pedigrees. Sometimes you're going to be given a pedigree, and a pedigree is just a family tree that looks something like this. And there may be another family over here. All right, and the circles are females, the squares are males, and typically there is a some sort of disorder that's being tracked. And I don't know what it is. We'll say that it's hemophilia, not hemophilia. We'll say that it's just blue eyes. It's being tracked. and blue eyes are recessive to brown. And all of these shaded squares and circles have the recessive trait. All right, so we can go ahead and figure out that all of these people are homozygous recessive because they have the trait. Now, what do we know about the other people? We know that Mama here does not actually have blue eyes because she's not shaded in, but she is able to pass blue eyes to the next generation. So she doesn't have blue eyes, but she is able to pass it. We would call her a carrier for blue eyes. So she has a daughter that's also a carrier. How do we know she's a carrier? Because dad only gives away blue eye genes. So this daughter also. Well, we would see the same thing down here. And so this was a relatively easy pedigree. But pedigrees are often going to show themselves. You may see a actual pedigree, or you may be asked to construct one. I would think if you're asked to construct one, it'd be fairly simple.